Hi, this is Nasa Sareen. We were dealing with metabolism, wherein we were dealing with glycolysis, where glucose gets converted to pyruvate. We have also seen the metabolic fates of pyruvate, where pyruvate gets converted to acetyl-CoA in presence of oxygen. This acetyl-CoA enters TCA cycle with enormous and gives out enormous amount of energy via oxidation processes. In the oxidation process, instead of getting ATP directly, you get the reduced coenzymes, that is NADH plus H plus, which gives you 3 ATP, and FADH2, which gives you 2 ATPs. This reduced coenzymes have to be converted to the energy currency ATP. How exactly we are going to do that? That is via Electron Transport System in our video 5. In electron transport chain, as the name indicates, it is the transport of electrons that takes place. Why exactly there should be transport of electrons? Because this transport of electrons gives shower of electrons. Via this there will be an enormous amount of energy. That is what we are interested. Whenever you eat anything, whether it is glucose or whatever, any energy-rich substrate, whether it is carbohydrates, fatty acids or amino acids, it has to break down finally to carbon dioxide and water. In the previous metabolic pathways, whether it is glycolysis or the intermediate step between glycolysis and TCA cycle or TCA cycle, we have seen that it is not going to give the carbon dioxide and water as the end product. If you start from glucose, it is giving you pyruvate via glycolysis. Pyruvate to acetyl-CoA, intermediate step. This acetyl-CoA condenses with oxaloacetate and enters Krebs cycle. Even in the Krebs cycle, you are not getting energy directly. You are getting the NADH plus H plus or FADH2. That is reduced coenzymes. You are not getting ATP. Neither you are getting energy, nor you are breaking down the complete breakdown of the glucose to carbon dioxide and water. If this has to be proceeded, that is carbohydrates, fatty acids or amino acids have to break down to carbon dioxide and water, then that cycle is via ETC. Suppose you have an energy-rich substrate. Okay, you have a substrate which is, if I consider it as A, this substrate has a reducing equivalent, okay, H2, which is a reducing equivalent. This substrate with its reducing equivalent is being transferred to the coenzyme. The NAD and FAD are coenzymes. So the H2, that is the proton, and the electrons are being transferred to the coenzyme NAD plus or FAD to change it to reduced coenzymes. The NADH plus H plus and the FADH2 are otherwise called as reduced coenzymes. These reduced coenzymes now have enormous amount of energy. That is NADH plus H plus has 3 ATPs. FADH2 has 2 ATPs. What has happened? The carbohydrates, fatty acids or amino acids, they are energy-rich substrate. Suppose whatever food you take, that has energy in it. This energy has to completely break down into carbon dioxide and water. In this process, whatever the metabolic substrate is there, it is having a reducing equivalent. This reducing equivalent is transferred to coenzymes like NAD plus and FAD. From the coenzymes, when it accepts a proton and the electron from the substrate, it forms reduced coenzyme. The coenzyme is no more a coenzyme, now it is reduced coenzyme. This reduced coenzyme will have ATP either plus 3 or plus 2 respectively with respect to NADH plus H plus and FADH2. The reduced coenzymes are not direct energy currency. If I would have got direct ATP, then there is no problem, like glycolysis, pair phase, glyceraldehyde 3-phosphate to 1,3-diphosphoglycerate. In that, you get energy plus 3 ATP, but it is in the form of reduced coenzyme. You don't get direct energy. Where do you get direct energy? Phosphoenol pyruvate to pyruvate, the last step of the payoff phase, where you get direct energy. That is, you get ATP directly. But here you are getting energy in the form of reduced coenzymes. So I have to manufacture or I have to get back the energy in the form of ATP ultimately from these reduced coenzymes. So what I do is I just move this reduced coenzyme and make them to enter the ETC cycle where they accept a proton and the electron, wherein they can react with the molecular oxygen and form water. That is what we are looking for, the end product as carbon dioxide and water. So they can react with molecular oxygen to form water, and during this process and there will be lot of loss of free energy. Little free energy is being that is the outer membrane, inner membrane, intermembrane space, cristae and matrix. Okay. The mitochondria has 5 distinct parts: outer membrane, inner membrane, intermembrane space, cristae and matrix. If you see, this is the outer membrane, this is intermembrane space. You can see cristae, matrix and the intermembrane space and inner membrane. Okay, mitochondria is made up of outer membrane, inner membrane, intermembrane space, cristae and matrix. The outer membrane supports the mitochondria. However, the main 3 components of mitochondria are inner membrane, the cristae and the matrix. In that, the inner membrane plays the vital role. The inner membrane has the ETC assembly that stands from here to here. This is known as ETC system or ETC assembly. Apart from that, it also has ATP synthesizing system. ATP synthesizing system. The inner membrane has the two important features. One is ETC system or ETC assembly and ATP synthesizing system. In this, the inner membrane is folded inwardly in the form of cristae. This cristae actually increases the surface area of the inner membrane. This inner membrane is also having phosphorylating subunits. Phosphorylating subunits. These phosphorylating subunits are the places where exactly they are specialized units. They are lollipop-like structures. These specialized units where actual ATP synthesis takes place. The phosphorylating subunits are the specialized structures. They are in the shape of lollipop where the actual ATP synthesis takes place. Apart from that, the inner membrane is folded in the form of cristae to increase the surface area. Apart from that, the inner membrane also consists of 2 important features. One is the ETC system and the other one is ATP synthesizing system. You will have to know that the inner membrane do not allow any kind of molecules, small molecules like ADP or ATP to enter the mitochondria. Not only that, it does not allow even the cations like H+, K+, Na+ to enter. So it is impermeable to cations or the smaller molecules. Further, the matrix is very important because this matrix consists of enzymes which hold good for any kind of oxidation reactions. Because via the oxidation reactions, we get the reduced coenzymes which have energy. So the matrix, whether you go for beta oxidation pathway or you go for TCA cycle or any amino acid synthesis, whatever enzymes are required for the oxidation process, those enzymes are provided by matrix. So mitochondria is appropriately called as the powerhouse of the cell. So this is the structure of mitochondria depicting ETC, F0, F1 subunit and the phosphorylating subunits, that is the complete ATP synthesis. How exactly the ATP synthesis takes place, how exactly the electrons are being transferred in the electron transport system, that we are going to deal in detail via multi-protein complexes. The multi-protein complexes in ETC. This diagram is a schematic representation of multi-protein complexes in electron transport system or electron transport chain. We were interested in the reduced coenzymes. The reduced coenzymes are actually derived from the substrate. What we said, there is one metabolic substrate which has the reduced equivalent. This metabolic substrate can be glyceraldehyde 3-phosphate, it can be pyruvate, it can be alpha-ketoglutarate, malate, succinate, whatever. This substrate consists of a reduced equivalent. This reduced equivalent is being transferred to the coenzyme NAD+. The coenzyme now gets converted to reduced coenzyme after accepting the reducing equivalent. So this reduced coenzyme now has ATP in the indirect form. How exactly this indirect form of ATP you are going to get in the direct form. That is, from any substrate, whatever you get, NADH plus H plus, the reduced coenzyme, this enters into the complex 1 of the electron transport system. In electron transport system, you have 5 complexes. This is complex 1, complex 2, complex 3, complex 4 and the complex 5. The 4 complexes are involved in the transport of electron. The 5th complex is involved in the synthesis of the ATP, that is energy. So, in the complex 1, it is otherwise called as NADH coenzyme Q reductase because you have some mobile carriers called as coenzyme Q. In the electron transport system, you have 5 complexes, that is, they are also involved in the carrying of electrons. Apart from that, the mobile carriers, there are 4 mobile carriers, they are also involved in the carrying of the electrons, that is coenzyme Q, FES, Cytochrome C, oxygen. These are the 4 mobile carriers. So you have complex 1, complex 2, complex 3, complex 4 carrier of electrons coupled with mobile carriers like coenzyme Q, Cytochrome C, oxygen, FES, the mobile carriers of electrons coupled with the complex 5 which is majorly involved in the synthesis of protein. Now starting on with the complex 1. The complex 1 is called as NADH coenzyme Q reductase because the substrate is getting the reduced coenzyme NADH plus H plus, hence this is NADH. The mobile carrier here is coenzyme Q reductase. So in this process, the NADH coenzyme Q reductase is a multi-enzyme complex which has FMNH2 as its prosthetic group. This FMNH2 has heme in it. So the electrons move from the NADH plus H plus to FMNH2 and via iron sulfur proteins it moves to coenzyme Q. Let us know what are iron sulfur proteins later. Let us deal with the second complex. The second complex where the substrate is succinate which is outside the ETC cycle. Whatever substrates are there, from succinate you get a reduced coenzyme FADH2. Here the reduced coenzyme was NADH plus H plus which is having 3 ATPs. From here succinate you get FADH2 which has 2 ATPs. So from succinate outside ETC you get the reduced coenzyme FADH2 which also gets from where the electrons get transferred to coenzyme Q, the mobile electron carrier via FES, iron sulfur proteins. This multi-enzyme complex is otherwise called as succinate coenzyme Q reductase. Succinate coenzyme Q reductase or NADH coenzyme Q reductase. So whatever the electrons are from either via the ETC or Krebs cycle or outside the ETC, they actually come to the mobile carrier coenzyme Q. So coenzyme Q is the actual mobile carrier which binds complex 1 and complex 2. There are the iron sulfur proteins. There are like half a dozen of iron sulfur proteins in the ETC system that are being discovered, but nobody knows their actual role. But however, it is told that one iron sulfur protein actually transfers the reduced coenzymes to coenzyme Q. So one iron sulfur protein transfers the reduced coenzymes to coenzyme Q. The other iron sulfur protein is being involved between cytochrome B and cytochrome C1. Now, since coenzyme Q is a mobile carrier which is of highly important in ETC, let us know what is coenzyme Q. Coenzyme Q is a mobile carrier of electron which is otherwise called as ubiquinone since it is ubiquitous in nature. Ubiquitous means it is universal in nature. That means any mammalian tissue will have coenzyme Q in its body. So this coenzyme Q is important carrier of electron. It is otherwise called as ubiquinone because it is a derivative of quinone. It is otherwise called as isoprenoid. It also has 10 isoprenoid units. So it is otherwise called as coenzyme Q10 because it is made up of 10 isoprenoid units. So coenzyme Q is ubiquitous in nature. Coenzyme Q is universal. Coenzyme Q is present in all mammalian tissues. It is a mobile carrier of electrons which carries electrons from the complex 1 and complex 2. And further it is called as coenzyme Q10 because it has 10 isoprenoid units. So coenzyme, there are only exceptional species, that is Mycobacteria, where coenzyme Q is not there. But if ETC has to take place in Mycobacteria, instead of coenzyme Q, vitamin K performs this function of coenzyme Q. So coenzyme Q becomes very important mobile electron carrier to enter the complex 3. Now the complex 3 has cytochromes in it. Let us know what are cytochromes. Cytochromes are conjugated proteins. So these cytochromes, they are called as conjugated proteins. They are colored proteins where the heme group, the coenzyme Q becomes the central mobile electron carrier because the complex 1 has the electrons which it is being passed to coenzyme Q. Even the electrons via FES, they are passed to coenzyme Q from the complex 2. So the electrons from complex 1 and complex 2, they are passed to the main mobile electron carrier, that is coenzyme Q. Now from here, the electrons are being transferred to cytochromes. Let us know what are cytochromes, which is actually the third complex, coenzyme Q, cytochrome C reductase. Coenzyme Q, cytochrome C reductase. So we'll have to know what are cytochromes. Cytochromes are actually conjugated proteins. Cytochromes are colored in nature. They have porphyrin, they have the porphyrin group which has porphyrin ring and iron in it. The heme of the cytochromes is different from the hemoglobin or myoglobin. The heme which you see in hemoglobin or myoglobin, Hb or Mb, this heme has the iron in Fe2 plus state. This is known as the reduced state, which is otherwise called as ferrous state. So the heme group has iron. The heme group of the hemoglobin or myoglobin has iron in it. That iron is always in the Fe2 plus, which is otherwise called as reduced state or otherwise called as ferrous state. Ferrous iron. Okay, but the heme group of the cytochromes, they have iron in the form of Fe3 plus or Fe2 plus. If it is Fe3 plus, it is oxidized state, and if it is Fe2 plus, it is reduced state. Okay, so either it is in ferric form or in ferrous form. So what is the funda of having this kind of a special heme group which has iron in the ferrous as the ferric as well the ferrous state? Here what happens is the ferric changes to ferrous, ferrous to ferric, ferric to ferrous, ferrous to ferric. Oxidation, reduction, reduction, oxidation. Because of this, there is oxidation reduction potential which takes place, giving out shower of electrons. That is what we are interested in. These shower of electrons ultimately to enormous amount of energy. That's why the cytochrome which has the heme group, which has the iron in ferric and ferrous state, becomes important in complex 3. The cytochromes initially were A, B, C. Cytochrome A, cytochrome B, cytochrome C. There were only 3 cytochromes that were discovered. But in electron transport chain, you see 5 cytochromes. That is cytochrome B, cytochrome C1, cytochrome C, cytochrome A, and cytochrome A3. Okay, cytochrome A and A3 are collectively called as cytochrome oxidase. From cytochrome B and cytochrome C, the electrons get transferred to cytochrome C. Like coenzyme Q, cytochrome C is also a mobile electron carrier. This consists of 104 amino acids in it. Of course, like cytochrome C reductase, this cytochrome C also has heme group in it. The heme group obviously has the ferric and ferrous state. Of course, there will be shower of electrons in the cytochrome C also. One thing to be noted here is cytochrome C is loosely bound to the ETC. Because it is loosely bound to the ETC, it's easily extracted extracted. If one has to study the entire ETC, how it is being proceeding, you can extract this cytochrome C easily. You can remove from that and you can study. So it is loosely bound. From the cytochrome C, the electrons move to the next complex, that is cytochrome oxidase. Cytochrome A, cytochrome A3 are collectively called as cytochrome oxidase. So how is this cytochrome oxidase different from cytochrome C reductase? Cytochrome C reductase has a heme group which has iron in ferric and ferrous state. Of course, cytochrome oxidase also has a heme group which has in ferric and ferrous state, the iron of which is in ferric and ferrous state. Apart from that, it also has copper, which is Cu2 plus and Cu plus. It is also in cupric and cuprous state. Not only because of the iron of the heme, there will be oxidation reduction potential. Because of the copper also, there will be oxidation reduction potential. If only iron can give you shower of electrons, iron plus copper can give you more electrons in it. So this is one of the importance of cytochrome oxidase. The second important thing of cytochrome oxidase is it is the only multi-enzyme complex which can react with molecular oxygen to give you water, because we are interested to get the end product as water. So cytochrome oxidase is the only multi-enzyme complex which can react with molecular oxygen to give you water. So we have seen till now from complex 1, complex 2, complex 3, complex 4, we have got shower of electrons, and there are coenzyme Q, ATS, cytochrome C and molecular oxygen where they act as mobile electron carriers. Via all this process with the complexes and mobile electron carriers, there will be enormous amount of energy that is being formed now. This energy from all the sides, from this complex, from the next complex and from the next complex, they all gather into the fifth complex, which is having an enzyme ATP synthase. We all should know that if your mom is going to give you like one apple, you are going to get an energy of one apple. Suppose if one apple is going to give you 1 ATP, so obviously if you eat one apple, you are going to get 1 ATP. Suppose if your mom gives you 10 apples, that doesn't mean that you get 10 ATPs. Okay, so our body will actually take energy, or it forms energy how much it is being required. So more if you eat, that doesn't mean that you get more energy. The same way there will be loss of energy. So if your body is mechanized to accept only one apple energy, it is going to accept only one apple energy. So irrespective of the shower of electrons from the ferric or ferrous state, or from the cupric or cuprous state, or from the reduced coenzymes, mobile electron carriers, whatever is there, the ATP synthase will pull out only so much energy that is required for it to change ADP plus Pi to ATP. So during electron transport system, there is enormous amount of energy which is being lost. Little free energy is being trapped by the ATP synthase to form ATP. That is what we are interested. The reduced coenzymes NADH plus H plus or FADH2 which were there, now they have given us direct ATP. So this is a schematic representation of electron transport chain where you get the end product of water, carbon dioxide and of course ATP.